Hello everybody and welcome back to another RevWise video. Today we're going to be taking you to the mechanism behind the polymerase chain reaction, otherwise known as PCR. This guide is specifically aimed for the IB Biology syllabus, the new syllabus with first exams in 2025. Now before we go into the actual mechanisms, it's important you know what PCR is used for. PCR is used to amplify small amounts of DNA into a much larger amount, a much larger quantity. Now, the reason we use PCR is often for use in DNA profiling. Now, the reason we need it for profiling is because when we obtain a DNA sample that we want to profile, it's often in a small quantity, often a swab of DNA from a crime scene. And in order to identify the identity of this DNA, we need to conduct gel electrophoresis on it. But gel electrophoresis can only be conducted if we have a large adequate amount of DNA. And in order to generate that large amount of DNA from the small sample we have, we need to amplify the DNA, and we do that by using PCR, the polymerase chain reaction. Okay, now that we've got that sorted out, let's just go into the mechanism. So I've done a bit of the stages here, but I want to take you through it, so I'll also draw the rest of it as we go through it. So in stage 1, we can see we have our DNA with the 5' and 3' ends labeled. So what can we see here? We can see that the temperature is being heated to 90 degrees, which in stage 2 causes the two strands to separate. Now this first stage where the temperature is heated to 90 degrees, causing the separation of the two strands, this is called denaturation. The high temperature causes the breakage of the hydrogen bonds holding the base pairs between the DNA together, and this allows the two strands to separate. Now, as you can see by the way I've drawn this, there are 3 stages, so remember the first stage is denaturation. It's going to be an important part of your answer when you talk about PCR. So after denaturation at 90 degrees, and also another note, the way that this temperature is achieved is due to the fact that PCR is conducted in a machine called a thermocycler, which uses temperature variations to basically manipulate each stage, to kind of start each stage, because each stage, stage 1, stage 2, stage 3, you'll see they each need specific temperatures, and through the thermocycler we're able to manipulate the temperature so that each stage can be conducted at the right time. Okay, so right now we have split our double-stranded DNA into 2 single strands, and let me just write the ends here. That's going to be our 5' end, our 3' end, our 3' end, our 5' end. Now, what happens next? Well, our next stage is called annealing. Now, what happens in annealing? It's pretty simple, but essentially the sample is cooled to roughly 55 degrees. Remember, this is in Celsius, Celsius. So from 90 degrees, we're cooling it to 55 degrees. But what is the purpose of this? Well, the purpose is for primers to anneal. Remember, the stage is called annealing, so it's pretty easy to assume that the goal is for primers to anneal. So if I were to draw the way in which primers anneal, it would essentially be at the 5' end of each of these strands, we would have our primers, and our primers are not just like that, though. Our primers also have their complementary base pairings like that, and we'll put it for this one too. This is essentially how the primers will look. So we have our primers with the complementary base pairs, and they anneal to that strand. And this is essentially what can what allows the next part to happen, And the next part is the main part that generates the desired effect that we want, where we want to amplify large amounts of DNA. The next part is called elongation, and I think you can guess what this one is. So remember, these primers are very important because we need the primers to anneal so that elongation can occur. Now, what happens in elongation? Well, if you studied DNA replication, you might have an idea of what kind of enzyme would be responsible for continuing these strands. Because in DNA replication, the one we use is DNA polymerase 3. That is what adds free nucleotide triphosphates, attaching them onto the leading and lagging strands. That's the mechanism for that. Now, in this, you might think we might use the same enzyme, but actually we use a different enzyme. We don't use DNA polymerase 3, but we do use a polymerase enzyme. And that enzyme is fundamental that you know it. So before I tell you the name of that enzyme, I'm going to just tell you what temperature, because it is pretty important and it's pretty interesting. So we have 72 degrees C for the next part to occur. Now, if I were to just take our two things here, okay? So you might think 72 degrees C. Why is that important? Well, if you think about it, 2 degrees Celsius seems quite high for an enzymatic reaction, right? Most enzymes in our body that we know have an optimum around that 37-degree range, right? So seeing a 72 degrees C, you might think the enzyme would be denatured, but this is where we need to identify the enzyme. See, the enzyme that we're using is called Taq polymerase. Now, why is this enzyme important? Well, firstly, it's what allows elongation to occur. It takes up a similar role to DNA polymerase 3 in DNA replication, but instead here, it's not with leading and lagging strands, it's just doing it to these two strands. It's just simply adding nucleotides to continue the strand and to successfully create two double-stranded pairs of DNA. Now, how is Taq polymerase able to withstand this high temperature? Well, it's all about the source. Taq polymerase is taken from thermophilic bacteria in the deep sea vents. And in these deep sea vents, temperatures are naturally very high. So as an adaptation, they have their enzymes have an optimum of a very high temperature. So if you were to justify why the temperature in the elongation stage is set to 72 degrees Celsius, you would say this is the optimum temperature for Taq polymerase to function. And if you needed some justification to why this is the optimum, well, Taq polymerase is derived from thermophilic bacteria in deep sea vents that experience very high temperatures, and so on and so on. Okay, next up, what happens? So we kind of can guess what will happen due to the fact that we know how complementary base pairing works, but I'll draw a few for you. So essentially, Taq polymerase will just be catalyzing this reaction. It'll be allowing this reaction to occur, and this will be allowing us to be adding base pairs just like this, really simply. And on both strands, this will be happening. Okay. So like that and that, and essentially this will just occur until everything is complete. Okay, until the whole we have two new we have two strands, we have one pair essentially of double-stranded DNA. So we went from one here to two here. Okay, and that is essentially the process of PCR. But wait, I know what you're thinking. At the start, I said this is the process to generate a large amount of DNA. But from here, I did double the amount of DNA, but it only went from 1 to 2. So how does this work? How am I going to get a very large quantity? Well, if you listen to what I was saying, you might have heard that each cycle of PCR doubles the amount of DNA. So when I went from one, I doubled it to 2. But the next cycle, it would double from 2 to 4, then to 4 to 8, then 8 to 16. And you can see how this would get very large very quickly. Now, how many cycles is run in a PCR reaction? Well, it's 30 cycles, important. And the calculation for the total amount of DNA generated is pretty simple, because since it's doubling each time, we just have 2 to the power of 30. That is the total amount of DNA we're going to get. So you can see how it gets very large very quickly, and that's just how PCR is going to happen. So that's essentially the process for PCR. It's very important not only for questions directly asking about PCR, but also in questions for asking about DNA profiling. And it's been very important not only in this, but it's good to have an understanding of PCR for other things you may experience in your life. Like you might have had a PCR test for COVID, and now you know the mechanism behind that. So yeah, hope you understood the video. If you have any other suggestions or any other requests for more IB biology or chemistry videos. I will do my best to upload them as I have lots of spare time now. Just let me know in the comments, and I will keep making videos for you guys frequently. So see you guys in the next one. Hope you enjoyed it, and see you guys later.